Hello everyone, welcome to my channel The Tooth Fairy, and our today's topic is camp reaction. So this topic is related to Streptococcus agalacti, which is a part of group B Streptococci, and it is usually asked as a 5 marks question for theory exams. Some other reaction which can be asked are Nagler's reaction or reverse camp. So CAMP is an acronym for the authors of this test, Christie, Atkinson, Munch, and Peterson. Its main use is to distinguish a group B streptococci, example Streptococcus agalacti, from other species of beta hemolytic streptococcus. So that means you're going to distinguish it from those bacteria which completely hemolyze RBC of blood agar. Now, other uses to identify bacteria, Listeria monocytogenes, which is responsible for listeriosis also. Principle of CAMP test is that group B streptococci, it produces a CAMP factor, which will act together with beta lysine of Staphylococcus aureus, that is a hemolysin that is going to break red blood cell. So CAMP factor along with beta lysine, they synergistically, they together cause more hemolysis. Now the types of procedures we do are standard test, disc method, and rapid CAM test or spot CAM test. The main method is standard test, which we will discuss in detail. If you see this culture plate of blood agar, here is a long streak down the plate of about 3 to 4 centimeter of Staphylococcus aureus, which has beta hemolysin, and then perpendicular to it there is another streak less, which is comparatively lesser in length than the main streak. Okay, यहाँ पर 2 mm का हम गैप छोड़ेंगे, ठीक है, यहाँ पे कॉलोनी यहाँ तक नहीं पहुंचेगी, ज्वाइन नहीं करेंगी ये स्ट्रीक्स आपस में. बट इफ यू सी हेयर, यहाँ पे बीटा हीमोलाइसिन था तो इसके नियर तो कंप्लीट हीमोलाइसिस होगी. ओके, but why there is an enhanced hemolysis here? It is because that this bacteria is group B bacteria and there is enhanced hemolysis. यहाँ पर देखें अगर इस साइड पे तो इस लेवल तक है hemolysis, but here it is till this level. So level of hemolysis is increased because of hemolysin and CAM factor synergistic effect. Then the procedure of standard CAM test, as we discussed, is then we have to streak a beta lysine producing strain of aureus down the center of a sheep blood agar plate. The streptococcal streak should be 3 to 4 centimeter in length, and then we are going to streak the test organism across the plate perpendicular to the aureus streak within 2 mm. We are going to incubate it at 35 to 37 degrees Celsius for like 1 day. Other two methods which we are going to read are disc test and rapid CAM test or spot CAM test. ये ऑप्शन है अगर लिखना है एग्जाम में या नहीं. Results we already discussed that if there is an arrowhead shaped or a butterfly shaped, then it's going to be a group B bacteria, and the negative is when there is no enhancement of hemolysis. So now in disc method, we are going to place disc which have some beta lysine, or that is hemolysin of Staphylococcus aureus, and then we streak microorganisms here, okay, at a distance of 2 to 3 mm. उसमें भी हमने मेन स्ट्रीक का टेस्ट ऑर्गेनिज्म वाली स्ट्रीक से हमने 2 to 3 mm रखा था, यहाँ पर भी रखना है. But the difference is that the positive result is a crescent or an arc shaped zone. That means जैसे मून होता है crescent shaped, अगर वैसी शेप आएगी then that is a positive result. In case of standard method, it was a butterfly shaped or arrowhead shaped. Next, we are going to discuss spot or rapid method, in which हम ब्लड अगार की एक प्लेट लेंगे, वहाँ पे एक ड्रॉप hemolysin, that is beta lysine, वहाँ पे ड्रॉप करेंगे, and near to that we will place Streptococcus agalacti colony. Then we are going to incubate it for 20 minutes at 35 degrees Celsius. Then you will examine the plate through transmitted light, and then if there is a zone of enhanced hemolysis, we it is a positive test. But we if we are still not able to see hemolysis, then we are going to re-incubate this for up to 30 minutes, okay, because the reaction was initially negative. But we do not keep it for longer period of time, because then it is going to give false positive test. So the limitation of CAM test is, we do not keep it under anaerobic condition. We keep it in ambient environment, because if it is kept under anaerobic condition, then even the group A streptococcus will be CAM test positive. Then, if we incubate for extended time or for elevated incubation temperature, हम 35 degrees Celsius पे कर रहे थे, firstly for 20 minutes, then for 30 minutes. But if there is an extended incubation time, then it can give false positive results. Also, the culture media used is blood agar, but उसमें sheep blood होना चाहिए. Human, horse, rabbit, or guinea pig blood plates will not give a proper reaction. So Streptococcus agalacti causes bovine mastitis in cows or buffaloes. That means inflammation of mammary glands, and it also causes various diseases in neonates such as septicemia, blood infection, brain infection, meningitis, and can also lead to septic, abortion, arthritis, osteomyelitis in neonates. It is otherwise commensal in female genital tract. That means normally it is not causing an infection, but if somehow infection is there, then it can be passed to neonates. This test CAMP reaction is basically the one of the diagnostic tests of S. galacti. Other diagnostic tests are Hippurate test and the sulfamethoxazole reaction. Okay, thank you. I hope this video was useful. Please like, share, and subscribe.